I'm going to start with a recap of previous last week, right? So if you weren't quite present mentally or physically or both, this would be a good time for you to follow so they can catch up, right? At the same time, it would be nice if you can run through with me. Every time I have little checkpoints where you can fill in with certain vocabulary, just run it through with yourself, right? You mouth it also good, right? It helps you remember things better. Okay? Alright, so a quick recap. Let's talk first about the structure of the mitochondria. The mitochondria has an outer membrane. It also has an inner membrane. This inner membrane is highly folded. Okay? So, outer membrane, inner membrane. Okay, quick recap for all of you. What is this space inside called? Sorry? Okay, good up. Just voice it out. Okay, so matrix. Next, what do we call this space between the outer and inner membrane? Okay, I heard something. Intermembrane space. Okay, so the intermembrane space. We need to know the structure as well as possible so that you can make sense about what's happening and where. Right? Other than that, outside the intermembrane space is the cytoplasm of the cell. Okay? Mind you, the mitochondria is not a cell. Okay? It is not a cell. Yeah? It is found within a cell. Okay? The cell makes use of mitochondria to get all the ATP it needs, which is the currency for energy within a cell. So now let's zoom in. Here I have a membrane. Here I have the intermembrane space, and this is the matrix. Therefore, this membrane is the which membrane? This is the outer or inner membrane? Inner. Okay? Can I follow it? So this is the inner membrane if I have to just make comparisons. We are zooming in now. Okay? Now let's go through all the stages of cellular respiration. Step 1 is called yeah, okay, thank you. Let's make our mornings together not as dreadful together. Okay, glycolysis, thank you. Glycolysis. If I were to break down the word, it's called glycolysis because glyco means sugar. Lysis means to break. So glycolysis. Right? In glycolysis, these are the things that are required. We start off first with glucose. Yeah? Glucose is broken down. Okay. What are the products of glycolysis? This is what's required of you, and last week we drew a table. Yeah? Okay, so quickly. Glucose, and then you get, thank you, pyruvate. Anything else you get as products? Sorry? ATP as a product, right? You get a little bit of ATP. Anything else you get? Thank you, NADH. This is a common thing of every step. Okay, so you forget everything, remember this. Every step you produce NADH. But what is the role of NADH? We learned last week the role of NAD plus. It is also known as an electron carrier because its role is really just to carry electrons. So if I were to draw this up, the role of NAD plus is this. It helps to take electrons out of glucose, and then you will get NADH in the process. Last week some students asked me, this equation makes 0 sense. Right? If you show this to Miss O, right, you also will find it very funny. How can the NAD plus electron you get NADH? Yeah? Yes? Oh yes, it takes place in cytoplasm. Drawn wrongly, So let me, how do I rearrange this? I need to shift this down a bit more. You know what? Let's box this up. This takes place. Thank you, Finya. takes place in the cytoplasm. Okay, last week, some students brought to me NAD plus electrons, how do you get NADH? Alright, so I'll just show you FYI. If you really want to balance things out. NAD plus 2 electrons plus H plus, you get NADH. Okay, this is the balance equation. If you really need to know. Okay, but you do not need to. Okay, for those interested. How is it you get NAD plus electrons, you get NADH? Okay, this is the balance equation. Okay, but you don't need to know, this is not chemistry. Okay, for bio, you just need to know that NAD plus can carry electrons. And when it carries it, it will be NADH. Okay, and moving on. So that was glycolysis. Takes place in the matrix. Okay, next. After glycolysis, next step is? Thank you. Okay, you're warming up already. Link reaction. For link reaction to occur, we need to make use of pyruvate. Okay, let's bring it into the intermembrane space. Let's bring it into the matrix. I'm going to go into the matrix now. Okay, so step 2. Link reaction. Pyruvate. Okay, what's needed and what is produced? Okay, what's produced? Let's go with what's produced first. Pyruvate, you get? Acetal. Okay, good. Acetal CoA. Okay, as a product. Anything else are products? NADH. Okay, see, it's a common thing. NAD plus will serve to take out some electrons again, and then you'll get NADH as a product. Any other products of after pyruvate goes through link reaction? Thank you. I heard CO2. Okay? Actually, now you're starting to see where CO2 is produced. Someone were to ask you, when you breathe out and there's CO2 in your breath, right? Where does CO2 come from? It came from specifically the link reaction in cellular respiration. You can actually draw the link now, exactly where it came from. Let's go to the last part of our revision. Okay, let's go to Krebs cycle next. This acetal CoA is then used for the Krebs cycle. And what do you produce from the Krebs cycle? Okay, in your notes, the Krebs cycle goes kind of like that. So this is acetal CoA. Okay, then there's like a cycle, right? A series of reactions. So what is produced as a result of Krebs cycle? Okay, so I have some other form of electron carrier. Okay, so there's FADH. If we get some electron out, it becomes FADH2. Okay, other than that, I believe you also produce some more NADH. So NAD plus, take out some more, you get NADH. Okay, anything else is produced in Krebs cycle? More ATP is produced. Anything else is produced in Krebs cycle? Last but not least, CO2. Okay, so I do not need you to memorize the steps as shown in your textbook. There are many steps. Just need to know the products at each point. Now, what's the purpose? Because you see, there will always be products that will be needed in the next step. Okay, each step is tied to each other by the products of the previous steps. That's why you need to know the products. of this takes place in the matrix, 2 and 3. While this one takes place in the cytoplasm. Okay, last but not least, last week we ended off with the last step called oxidative phosphorylation. In step 4. Just going to put it as, okay, let me write the whole thing. Oxidative phosphorylation. It is the last step. Okay, in this last step, previously I said step 1 to 3. If you notice, there's one similarity at every step. And that similarity is that we are using NAD plus to take more and more electrons. Right? At step 1, here we produce NADH. At step 2, we also produce more NADH. At step 3, we also produce more NADH as well as FADH2. They are all electron carriers, they are now carrying electrons. But for what purpose? For this particular purpose. Because at this final step, all of the NADH will release its electrons. Okay, and then you regenerate this NAD+. All the electrons will now enter this chain of proteins. So where am I at in your textbook? This page is very important. Yeah? Okay, to recap from last week we watched a quick video. Okay, this is what you are seeing on the whiteboard, right? So what happens to all the NADH? This is what happens at this final step. You see, NADH will donate all the electrons it's been storing. The electrons in this particular video is represented by the red color. The electrons will be donated by the NADH, and then it will be passed down the chain of proteins on the inner membrane. Watch as the electrons get passed down. Now let's look a little bit closer. Where do the electrons eventually go? It is finally received by the very end. Look at the very end on the right hand corner. The electrons will be received by oxygen. Okay? That's why you need to breathe in to receive oxygen into your body. Because oxygen serves as what we call your textbook a final electron acceptor. So may I get you to highlight that in your textbook? That's a very important vocabulary you can use to describe oxygen. Okay, oxygen is the final electron acceptor. We went through all this trouble just so that NADH can pass electrons down this chain eventually to oxygen. The word is bolded, in fact, over here. Yeah? All the electrons will eventually reach oxygen, the final electron acceptor. May I now also get you to look at the diagram provided to you? Can you find where this is happening? Right, in words they say oxygen receives the electron as the final electron acceptor. But in this diagram, can you see where that is happening? If you can, will you just give me a quick highlight so that you are aware? Where is oxygen receiving the electrons as the final electron acceptor? Okay, look at the extreme right over here. Oxygen, then they show one black arrow, they become water. Then you see this yellow arrow over here. What they're trying to show you is that the yellow arrow is showing you that the electrons are coming in, entering oxygen, and becoming water. Okay, so if you have not labeled it, this yellow arrow is representing the flow of electrons. Right? If you have not written that down, you may want to write that down, the flow of electrons. Now I'm going to spend a little bit more time over here so that you understand this as much as possible. This part of the chapter is the toughest part. If you understand, then I would say you're good to go. So we'll invest a little bit more time here before I let you all off. These three paragraphs pack a lot of vocabulary that's very new to you. I'm going to run through sentence by sentence so that you appreciate this better, and because in the next chapter you'll see almost exactly the same thing. Right? So it will be good time invested if you pay attention here. Alright, so let's listen up. Earlier on we said that we collected a lot of NADH, right? Okay, can you see the magnet? How you can see NADH? Okay, at every step we collect a lot of NADH. What does NADH contain? At the moment it contains electrons. I will represent the blue magnets as electrons. Okay? Every time you see a blue magnet from now on, I'm using it to represent electrons. Okay, so let's go sentence by sentence. During this final stage, electrons are transferred from NADH and FADH2 to oxygen, also known as a final electron acceptor, through a series of membrane proteins in the electron transport chain, regenerating NAD plus and FAD in the process. If you highlight, can you highlight the word electron transport chain? Okay. First, you may be wondering, what is the electron transport chain? Electron transport chain refers to all the proteins here. 1, 2, 3. These are proteins of the electron transport chain. We call it electron transport chain because it is a chain of proteins that transport electrons. Okay, so that's the first new thing of this paragraph. Following? Okay, let's go to the next sentence. As electrons are transferred from protein to protein, okay, so let's visualize this on a whiteboard. Okay, see following? Look up. Alright, so all the NADH we've been accumulating, let's bring them into the matrix right now. There's lots of NADH now. I'm going to erase this away so we have a little bit more focus. Okay. They say, let's look at proper. Electrons are transferred from NADH to oxygen through a series of membrane proteins. Okay, so this is what's going to happen now. Ready? NADH will donate the electrons and will become NAD+. Yeah? NADH will donate the electrons and become NAD+. Yeah? Okay, so that's what they're saying. Every time you donate electrons, you will regenerate NAD+. Then they say as electrons, second sentence, as electrons are transferred from protein to protein. Okay, as electrons are transferred from protein to protein, protons are driven across the inner mitochondrial membrane into the intermembrane space. Okay, what are protons? You may want to highlight the word protons. Okay, protons equals to H plus ions. Right? So you may want to indicate that down. I don't see a lot of writing. Okay, so protons refer to H plus ions. Okay, so continue the story on. Go back to sentence number 2. As electrons are transferred from protein to protein, from protein to protein, protons are driven across this is the inner mitochondrial membrane, driven from across the inner mitochondrial membrane into the intermembrane space. Okay, right now, this is the intermembrane space, this is the matrix. Every time electrons are transferred from protein to protein, some protons will be pumped from matrix to intermembrane space. This is what they're trying to say. Okay? You may be wondering, how come we need the electrons to be transferred? Then the protons can be pumped across. Well, I don't know if you recall our first time together studying mitochondria. We talked about how, when iron receives electrons, when iron donates electrons to oxygen, there's some heat energy release, right? Same thing's happening here. When an electron is passed from protein to protein, some energy is released. And these proteins make use of the energy to pump some protons across. So that's where the energy comes from. And therefore, this process of pumping the protons from the matrix to the intermembrane space is a form of active transport. I think we learned this in our previous chapter. Active transport requires energy. Okay? Maybe I'll write down what I was saying in words. Where does the energy to pump the protons come from? Okay? As electrons are transferred down the shortening of the word electron transport chain, the energy released is used to pump the protons. Okay, so this is where the energy comes from. It comes from the transfer of electrons. If you want to write it down. All good? Okay, so we're nearly done with the paragraph. Next word you want to use in your vocabulary is proton gradient. So you want to highlight this word, proton gradient. If you look at the whiteboard now, you will notice that there is a high concentration of H plus in the intermembrane space and a low concentration of H plus in the matrix. This is what a proton gradient means. When we talk about a gradient, we are saying that there is a high point and there's a low point, and this is the gradient. So what we are saying here is that now there is a proton gradient that has been set up, where the high is in the intermembrane space and the low is in the matrix. That's all it's trying to say. Okay, next paragraph. During chemiosmosis, the protons flow back down the gradient through ATP synthase. Okay, so this is what it means. Chemiosmosis. Chemi means chemical. Osmosis means the flow of something. So flow of what is the flow of these protons. During chemiosmosis, the protons flow down ATP synthase back into the matrix because there's a high and there's a low concentration. So facilitated diffusion is happening. As this is happening, ATP will be produced. I would like to show you now a video of how ATP, the enzyme is working, because it's a very fascinating enzyme. You only learn a little more about this when you go to a higher university. Okay? But I just want to show you a video because it's very cute. This is the enzyme. Okay, this enzyme over here is this one over here. What I cannot show you is this. Watch what happens as the protons flow through it. The protons are the green little dots. See, as the protons flow through it, it starts to turn. And every time it turns, the chemicals that are needed to make ATP can go into the active site. So it's almost like this. Have you ever seen online or on TV shows before a river stream? Then sometimes they put a wheel over there. And when the river flows past, the wheel will start to turn. That's what this enzyme is like. It needs the protons to flow through it so that it can get its wheels turning so that the enzyme can work. Every time the protons flow through, this enzyme starts to function. Then we can produce the ATP that we so want. Okay, and then you can see the things going ADP and phosphate, then put, and then you get one ATP coming out. Okay, so this enzyme requires the flow of protons. Yes? Okay, so very good question. You always ask me where do all these things come from. That's why we really need to eat. A lot of these raw materials to produce all these things come from the food we eat. Oh, this is not glycolysis, this is the last stage, oxidative phosphorylation. Okay? So this is how the enzyme works at the very last step. Okay? Finally, last paragraph. All in all, electron transport chain serves two important functions. It helps to establish the proton gradient, which is what we just talked about. It also helps to regenerate NAD+, so that the earlier stages can continue on. Notice over here, we produce NAD+, right? Where do these NAD+ go? Well, we can bring it back to the starting point. Let's bring it back to glycolysis so that it can be used again. Let's use this NAD+ for link reaction so that it can be used again. Let's use this NAD+ for Krebs cycle so that it can be used again. So we recycle a lot of the things we use within cellular respiration. Hi, you have a question? Scratching your face. Okay, so I will stop here for oxidative phosphorylation. I hope that you roughly understand this particular process. Okay, everyone can drink some water to refresh yourself. we take a mental 2-minute break. You can use this time to read through the paragraph, jot down some notes, consolidate your learning. Okay, we'll take a 2-minute mental break before we continue on. Okay, spend some time taking down notes, consolidating the notes. Yes. Oh, okay. So, as opposed to. Okay. So, question has been asked. Why are the protons pumped from the matrix to the intermembrane space, as opposed to just leaving it here like that, right? Okay. So, let me go back to this up. This enzyme here needs protons to flow through it. Let's assume that the starting point, there were already a lot of protons here. If at the start there's already a lot of protons here, then there's no need to. But the reality is, at the start, there are almost an equal amount of protons on both sides. There's no difference in concentration, so nothing will flow through it. That's why we need to pump it. We need to pump it just so that we can create a high concentration here, so that it will flow through this special enzyme. Yeah. Okay, as you consolidate in those 2 minutes, more questions may appear, can raise it up. Henry, got a question? Okay. Okay, before I move on to the next chapter, okay, we do a quick morning exercise. You raise your hand now. Okay, now I want you to do this action as fast as possible and see who lasts the longest. Okay, you raise your hand and then you do this. Let's see who can last the longest. What will you start to feel? Why are you all slowing down? Come on, go fast. Okay, this is as fast as I can go at this point in time. I'm starting to slow down. Now I want to go faster, so cannot go faster. Okay, I can roughly tell who are gamers here. Okay, I'm starting to slow down. Do you feel the burn? Oh, you still don't feel the burn? You are pretty strong, Okay. Mary, are you going at it deliberately at that speed, or you're a bit stuck already?
I don't know.
Do you feel the burn in your arm? Feel it? No, don't feel. Okay, then just stay and continue. Okay, your arm feels stiff now? It feels stiff? You know what you just experienced here? You've just created a situation where, first and foremost, for you to do this, your muscles need energy, right? And it needs energy in the form of this ATP that this enzyme has been producing. But what happens if you need a lot of energy, but there's not enough oxygen for you to produce it? For this last part, we're going to talk a little bit about anaerobic respiration. The word ana means don't have. Aerobic means if you never breathe. So, what happens if you do not have oxygen? Okay? There are two situations for this. One is you don't breathe, then you have no oxygen. Another would be what you just experienced. Your muscles require more energy than your rate of breathing. You're not breathing fast enough to supply oxygen to your arms. Yes? Yeah. Yeah. Right, okay, that's a good point. The reason why we still produce this is because this energy that's released, right, it is not something that can be used for chemical reactions. Okay? We produce this because this ATP can be brought anywhere around the body and then it can be used as a credit card to carry out any reaction. Okay, so the reason why we need to know the products and the process very well is because of this final thing. Look what happens if we do not have oxygen. Do you see the implications of the chain? If we do not have oxygen, this is the problem. Watch what happens, NADH comes by. It donates its electrons. You get NAD+. But as the electrons go on and on, it'll get stuck. There's no oxygen to receive it. This will what happen if you don't breathe. Another NADH comes. Ayo, stuck already. Another one may come. The electron transport chain, the ETC, will cease to function when you do not have oxygen, because you cannot pass electrons down anymore. So what will happen? This is a very major problem, because if I have another NADH coming, now it cannot release its electrons anymore. Yeah? So there will be a problem here. But you see, the bigger problem is this. Now, we will no longer have protons being pumped. No more protons flowing down. No more ATP being produced. So that's why you need to breathe. Okay? So that oxygen can serve as this final electron acceptor. But our body has a way around it. You felt that burn in your arm just now? That's because our body is producing something called lactic acid. You scroll down to the last page of your textbook. And if you look at this simple reaction over here, okay, our body can cope with a lack of oxygen for a short period of time. It does so by making use of step 1, glycolysis. Do you remember glycolysis can produce a little bit of ATP? Yeah, a little bit of energy. Well, in the event that we do not have enough oxygen, we have a backup plan. We will make use of glycolysis as the backup plan, so they can supply us with a little bit of energy. How, you may ask? Well, the pyruvate that's formed, our body will convert it to lactic acid. When we convert it to lactic acid, we end up producing NAD+ in the process. And this NAD+ can be used to allow other glycolysis to carry on so that we can get a little bit of ATP. Okay? So that's in the case of human beings. Right? If we do not have enough energy, we can make use of glycolysis. We'll produce lactic acid and a little bit of NAD+ so that we can allow other glycolysis to continue on. In the case of microorganisms like yeast, they don't produce lactic acid, they produce alcohol. And so the wine that your family members may drink, or the beer, that all occurs through the process of anaerobic respiration. move on to the next organelle, which is the chloroplast. Yes. Only in the case of microorganisms like yeast. So for human beings, we only produce lactic acid. We don't produce carbon dioxide as a byproduct. Sorry? Because this reaction I'm showing you is not the lactic acid one. It is the reaction above. Okay, look in the notes. You scroll up, there's one more cycle. Yeah. This one happens in yeast, not human beings. Okay. We're going to start to move on to the next organelle, the chloroplast. Okay, do you have any more questions before we move on? Yes? Yeah. Sorry, say again? It doesn't do anything, it's just a waste product. Okay? So, in the event that you do not have any oxygen, this is what your body can do. Our process of glycolysis goes like that, right? Okay, so our body will do one more step. Let's convert this to lactic acid. Remember earlier, we always have this step over here, right? NAD plus will take out electrons and become NADH, right? Yeah? Okay, so when pyruvate is converted to lactic acid, the NADH that was produced will be reused to produce lactic acid. And then you will regenerate this NAD+, then it can be used again for glycolysis. But not every time glycolysis occurs, you will produce a little bit of ATP. Your next set of notes, rather CA 3.5. Okay, there's a demonstration. But before I begin with the demonstration, you know what? Let's begin the demonstration. Anyone wants to volunteer for demo? You will not get hurt. Okay, you're ready? Okay, if you think you're ready, may I get you to open up CA 3.5? You should also have your notes for chloroplast on standby, because I may refer you there whenever I think there's something worthy for you to highlight or take down notes for. Now we're going to go into chloroplast. Sorry, you all know what chloroplasts are from primary school? Okay, you can find them in plants, yeah? Okay, so I hope that's the baseline at least you know. What's inside chloroplasts? Chlorophyll, thank you. Chlorophyll. There's the pigment that helps to trap sunlight. Okay, and that's what we're going to talk about today. Biology, there's a lot of chemistry involved. Okay, before I show you this demonstration, which I find quite amazing, I'm going to talk a little bit about chemistry. I'm sure you've seen this diagram before, right? You've seen, for example, this is the nucleus of an atom, and then often you have orbitals of electrons, right? Yeah, I'm going to draw orbitals of electrons. Okay, I just need to let you know, whatever I'm showing you is a simplified explanation, right? If you show this to Miss O, maybe she will flip in her grade. Yeah, so electrons. Okay, remember this diagram? Yeah? Okay, so the reason why I'm showing you this diagram is because it's something quite interesting and fascinating. Electrons kind of exist at their own energy levels or orbitals, right? However, there is a way that we can make electrons gain enough energy so that they jump to another orbital. Let's say, for example, I provide this electron with enough energy. Something interesting happens. The electron will become excited. It will become so excited that it will jump to the next energy level. Okay, so point, jumps. I would say that this electron is in its excited state. The original level, I would say, is in a ground state. Yeah? But this doesn't last very long, right? If I stop supplying it with energy, right, it won't stay in its excited state anymore. Instead, it would go back down to its ground state. But this is where the magic occurs. When it goes back down to its ground state, the energy that was supplied to the electron will be released into the environment. This can be released in the form of heat, will be released in the form of light. Two of the examples. One such atom that is able to do this particular interesting phenomenon is called quinine. That I'm about to show you. Quinine, if you supply with just enough energy, its electrons can be excited to the excited state, and as it goes back down to its ground state, it will release energy. And I'm about to show you what happens when it releases energy. Have you ever seen this string before? Have you not? Never seen before? Okay. If you go to 7-Eleven or go to NTUC now, or not now, after school, you'll be able to find this drink. It's called tonic water. Anyone likes to drink tonic water? Anyone drank tonic water before? Never. Okay, later on, you can have this can, whoever will demo for me. Okay, then you can try the drink yourself. Tonic water, and on the can it says contains quinine. Okay, I found this very interesting. I was like walking along the aisle, and it contains quinine. Okay, that means it can perform this particular reaction. Okay. Here, I brought a high energy torch. You know the UV light that comes from the sun rays? Yeah, I bought a torch that just shoots out UV light. It's very powerful UV light. Shine on your skin too long, you get cancer on your skin. Okay, so this is UV light. Doesn't quite seem like there's anything. Yeah? Unless it strikes certain surfaces. But you don't quite see anything coming out from this torch. That's because the UV light that's coming out is colorless. Right? By right, you cannot see UV light. You can see other forms of light, the visible spectrum, but not UV light. So high energy. Okay, anyone want to do the demo with me? Okay, come. Okay, let's invite Shaka. Round of applause for her. Okay, so how about you handle the quinine? Okay, ready? Okay, here inside this can is quinine, right? So Shamika, can you open up the can? Right now, I'm just I'll pour a little bit of quinine out. If we pour a little bit of quinine into this can, glass bottle, okay, thank you. That will do. Nothing quite special, right? Okay, but there's quinine in here. Okay, now I'm going to show you something interesting. Oh yeah, let me throw this away first. Let's supply the quinine with high energy. Okay? Okay, ready? Okay, you aim into this can, this glass bottle. Ready? Okay. Now you pour. Pour some more, pour some more. Alright, thank you. Okay. Thank you so much. Okay, round of applause, Shabika. Okay, you go back. Oh yeah, here's your drink. Okay, you can have the drink. Thank you. Okay, just for FYI, the drink doesn't taste very nice. Quinine, it's as a chemical, its electrons can be excited. When they get excited, you go to its excited state, right? As it's supplied by these high energy UV rays. But as it's also going back to its ground state, you release the energy. But interestingly, it releases the energy in the form of light. Yeah. If I don't supply it any energy, then there's no more, it won't get excited anymore. And it will not release any more light. But if you supply it enough, then you'll get this effect. There are other chemicals inside your pencil box that can achieve this same effect. Your highlighter also can. If you take your highlighter now, and if you were to just draw onto your paper, and I go around shining it up, you also see glow in the dark. The paper that you see on my whiteboard also contains chemicals that can glow when supplied with enough energy. Other things will be your highlighters. Okay. The reason why I'm showing you all this is because chlorophyll has this same effect. Okay, the chlorophyll pigment. And went through the trouble of extracting out a vial of chlorophyll. Okay, so here I have chlorophyll from plants. And here is high energy light. Let me guess what color this will glow. Green. Okay, I'm going to shine it up. The effect won't be as mind-blowing as what you just saw, but you will see a color. Ready? Okay. I need to shine it in a way such that I don't get skin cancer. Every time after I do this trick, right, then my fingers will feel very tingly. Do you see a color? See it? Yeah? It glows a blood red when I supply it with energy. It glows a blood red. Okay? However, if you notice, when you look outside, you don't see all the leaves glowing red, right? Even though there's UV light striking it. Right? So, why? Why don't we see leaves outside glowing red, even though there's constantly UV rays striking it? Sorry? That could be one thing. Maybe it's not strong enough, maybe it's not concentrated enough. Another thing could be that something is happening here. Right? The only reason why we see the color, the light, is because energy is released as it's going back down to its ground state, right? So something must be happening here. Well, that's exactly what the plants are doing. Rather than allow the energy to be released in the form of red light, the plant uses this energy to produce something useful. Okay, that's what our chlorophyll pigments inside the plants are doing. Right? It captures the UV light that's coming in, but it doesn't allow the energy to be released for useless purposes. It uses that energy for a more useful purpose. And that's why we study the process of photosynthesis. Because photosynthesis is really about using light, which is photosynthesis, to produce. We are using light to produce something useful. Right? And this is what we're going to do in the next chapter. Whatever I just said, you may find it in this part of the textbook. I'll give you some time now to jot down some notes if you like to. Okay, whatever I just went through was this little part of the textbook. Just a quick demonstration for you. Just as we did for the previous chapter, let's start off first with the structure of the chloroplast. Okay, so that you are a little bit more aware of the terminologies I'm using. So biology is a lot of it is part of the vocabulary, knowing how to string them into a sentence to make sense of it between biologists. A lot beyond. Okay, let's look at the structure of the chloroplast to start off the lesson. I'm currently on page 41 to 42. Okay, 41 to 42 is where you can find the relevant information I'm going to be talking about. Okay, I'm going to zoom a bit up. We know that in mitochondria there's an outer and inner membrane. The chloroplast have the same thing. Okay, so we start with this. The chloroplast is an outer membrane. Very often chloroplasts look a little bit like M&Ms under the microscope. Chloroplasts also have an inner membrane, but here's difference number one. The inner membrane is not highly folded. Like M&M is very thick, full of chocolate. Okay, there's also an inner membrane, but not highly folded. So there's one layer, and two phospholipid bilayers. The chloroplast, however, is special in that it has a third layer of phospholipid bilayer. Okay? We have this third layer of membrane that is highly folded. And it's highly folded into very cute little pockets, or look almost like pancakes. Okay? This third layer is folded, and then it's folded to look like little pancakes. It's just a side view. Okay? If you look into your notes, you will see that another view of it. This third layer of membrane, we call it the thylakoid membrane. Okay, so this is number 3. Yes?
Is that more of one of that kind?
Say again?
Is that more of one of like
Yes. So you will see in your often in the notes that there is a lot more of these structures present. A lot of them. Collectively, we tend to just refer to all of them as the thylakoid membrane. So the question was, are there more than one thylakoid membrane? You could say so, but I would say that it's just the third layer. I wouldn't say that because I have 4 thylakoid membranes, I have 4 layers in total. Right? It's just one layer. Okay, other structures that are relevant to you. Okay, this space over here. Okay, this space over here where the thylakoid, all these things sit in, is called the stroma. Okay, it's called the stroma. Next, inside these stacks, right? The space inside these stacks are called thylakoid space. Right? There's intermembrane space. This is the thylakoid space. in your textbook, they use the word lumen instead of space. That's also fine. Lumen is the biology way of saying space. Okay, so thylakoid space or thylakoid lumen, these are all fine. Okay, I just need you to know these following structures to appreciate how the chloroplasts work. Outer membrane, inner membrane, thylakoid membranes, thylakoid space at inside, as well as the stroma over here. The chloroplasts also have intermembrane space, but this is not relevant for this chapter. Okay, we rarely make mention of it, so you can ignore the intermembrane space because it's not very useful for this chapter. Yes.
Where's the chlorophyll inside?
Okay, so where do you find chlorophyll? Is it free floating around? It's not. You'll find chlorophyll on the membrane of the thylakoid membrane. Okay, this is where you find all the chlorophyll pigment. The chlorophyll pigment is not floating around. It is also not on the inner membrane or outer membrane. It's all in these small little hanging steps. Yes.
Are the chlorophyll like the ribosomes? Like, are they on top of the membrane?
Okay. So, now even deeper question. Are the chlorophylls just let's say this green circle is chlorophyll. Is the chlorophyll, and let's say this is the thylakoid membrane. Is the chlorophyll like here? Or is it on the surface? Or is it on the bottom? Right? That's a great question. Okay, this is where you find chlorophyll. Chlorophyll is actually arranged and organized in a protein structure. And this protein structure looks something like that if I slice it into half. Okay, this structure, you can find it on a subsequent page of the textbook. Over here. Here. Okay, this is where you can find chlorophyll. Which just so happens to be the next part of our chapter, yeah? Okay, so the question asked by Hiran is, where? Where do you find chlorophyll? This is where you find it. Number one, you find it on the thylakoid membrane. So that means over here, right, the thylakoid membrane. Number two, the chlorophyll is actually found within these interesting looking protein structures. And this structure is called, okay, inside your textbook, they label it as a photosystem. Photo means light. System means that there are many parts. It's called photosystem because this is made up of many parts. In fact, it's made up of two kinds of parts. You see this center region over here? This center region is called a reaction center. I don't know how to spell center. Complex. I believe it's all in your notes. A reaction center complex. The word complex means that it's complex. It's made up of many things. Okay, you do not need to know what it's made up exactly. Just understand it's made up of many things. It's called reaction center complex because this is where a reaction will occur. Next, you see structures surrounding this reaction center complex. Each one of those is called a light harvesting complex. It's called light harvesting because it harvests light. And this is where we find our chlorophyll. This one? Oh, yes, this entire thing is a chloroplast. Thank you. So, where do we find chlorophyll? We find it inside a photosystem, inside the light harvesting complex. So I would like to direct your attention to these white circles that are drawn inside the light harvesting complex. That is where you find chlorophyll. Okay? So if I use these green circles, this is where you find chlorophyll. Yes. Yeah. Okay. In this diagram on this page, you see the word. When you see the word pigment molecules, they are referring to chlorophyll. Okay. So you can go to this page, circle this, and write chlorophyll. Chlorophyll is a pigment. Pigment means what is pigment? Pigment is the thing that gives color. Okay, so chlorophyll is a pigment molecule. So now we know where chlorophyll can be found. Let's relate it to whatever experiment we just did. You know when we shine light, right? Then the electrons can get excited. When it goes back to its ground state, energy is released that was once absorbed. But where does the energy go in plants? So this is where it goes. Yeah, ready? So these are chlorophyll pigments, right? So I say chlorophyll pigments also have electrons, right? Okay, so now, remember say again these blue magnets? Okay, I'm going to overlay every chlorophyll pigment, I'm going to put electrons into it. Okay? Yes? Question? Okay. So, now, what will happen if we have light? Striking these chlorophyll pigments. Okay, watch what happens. The electrons will get excited. Okay, so this is the only way I can show this. I'm gonna show you that it's excited by making it jump out of the whiteboard. Okay. Okay, so light strike, it gets excited. Yeah, so it's excited now. So when it goes back to its ground state, I'm going to represent it by it just going back down to the whiteboard. Okay, so this is how I'm representing excitation. Okay, so let's try again. Light strikes, the electron gains energy, it gets excited, and then it goes back down. Okay, so now the question is, as it goes back down to ground state and the energy is released, where does this energy go? Okay, this is where it goes. Watch what happens. It gets excited, goes back down to ground state, it's releasing energy. But where does the energy go? Actually, it goes to the next chlorophyll pigment. Next chlorophyll pigment gets excited. As it is going back down to ground state, it passes the energy that is being lost to the electron in the next chlorophyll pigment. Then as it goes back down, it passes it to the next chlorophyll pigment. So essentially, have you gone to a soccer match or a match where people do this and they, whoa, then there's a wave of people? It's just passing energy in that sense. The electrons get excited, and then as it comes back down to its ground state, the next electron beside gets excited and goes back down to the ground state. That's where the energy is going. So if I do it all together, there's a light harvesting complex here, there's another one here, let's do it all together. Light strikes, they get excited, then they transfer the energy to the next electron, and the next, and the next. Okay? And And therefore, if you look at this diagram over here, you see these black arrows here? The black arrows represent the transfer of energy. Right? Is it written here? Okay, so you may want to highlight this. That's what I'm trying to show you. The black arrows represent the transfer of energy. Okay, all good? Okay, one last thing. So, where does the energy go from here? The electrons here get excited, and then as they go back down to ground state, my question is, where does the energy in this last chlorophyll pigment go? If you look at the textbook, it goes to the chlorophyll right at the bottom in the middle, the reaction center complex. Okay, this one over here. So you know what's happening? All the light harvesting complex surrounding it is channeling all of its energy to this single chlorophyll in the middle. Okay? So we do again, ready? Light strikes. Excited, 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 and finally, all of the energy is transferred to this electron here. But because of that, it gets so excited that it doesn't go back down to its ground state. It jumps out of its chlorophyll to the top of it. Jumps out of its chlorophyll into this part on top. You see in the textbook it says primary electron acceptor. Yeah? The chlorophyll over here gets so excited that it loses electrons to the primary electron acceptor. Jumps out of the chlorophyll itself. In a sense, now its chlorophyll is charged up. It's lost an electron. So why am I showing you all this? Well, the photosystem can be found along with the electron transport chain that you just learned in the cellular respiration. Remember, in an electron transport chain, what does it transport? It transports electrons down the chain. Photosystems can be found as part of the electron transport chain in the chloroplast. So this electron from here will go through the same process as what you just learned earlier on. From here, you will pass down an electron transport chain from protein to protein, and as it's being passed down, you do exactly the same thing. It will pump protons from one region to another. That's why you need to be very familiar with the electron transport chain. It can be found in plants and also in animals. Just that in plants, it doesn't get its electrons from NADH. It gets it from the photosystem. A very excited photosystem. Okay, I'm going to pause a while now for you to consolidate notes and for you to ask me any questions that you may have. how when you go to university level, you learn that between here to here, there will be a protein that will help to shuttle the electron to the next location. Okay? Then when you go to the university, you also learn that there will be another protein that will help to bring the electron to the next location. But I simplify it. All I just show you is that it jumps from here to here, then jump, then jump. Yeah? More questions? Yes. Question?
And how does the red fragment go from the top, the proton, how does it go from the top to the bottom?
Oh, how does it go from here to here, is it? How? Okay, so the same way as in the cellular respiration, the last step, every time electrons are being passed from one protein to another, some energy will be released in the process. And that released energy will be used by the protein to active transport pump it across.
Then from there it forms ATP.
Oh, so then from here, we do exactly the same thing. We accumulate a lot of protons, then it will flow down, then as it flow down, ATP will be produced. So it's
The electron pass on go until the special one with the reaction sequence.
here,
Yeah.
or the energy will be passed until it reaches this electron, and it will get so much energy until it will jump to this, then after it will get passed on.
So it's passed down that way first.
Passed down this way?
So if that's the end point, then it starts the right side, right?
You mean left side, you mean?
Wait, that's the end point, right?
No, this is not the end point. The end point is somewhere over here.
Then the end point has a special reaction center.
Yes. Okay, what do you mean by end point?
Like, which one is the one that has so much energy that it jumps out?
Oh, okay. It's over here. So if you look at this diagram here, you look at this liap over here with like, look like backside, right? Yeah. So the energy that's transferred to this one over here, you'll get so much energy, the electrons, that it will jump to here, and then after it can go down the chain already.
So that's the first one or the last one?
Say again?
So that's the first one or the last one?
I don't understand what it means. What you mean by first one or last one?
It's got to start somewhere, right?
Sorry?
It starts somewhere, right?
What starts somewhere?
This whole process.
Chain, the electron.
Yeah.
Oh, the electron transport chain. When does it start, is it? Oh, okay. So it starts from after this electron gets transferred here, then it starts going down the chain itself.
Then it goes back again.
Goes back to where?
Back one.
Back to here?
Yeah.
No, it doesn't go back. It will go down to the next protein. Okay, so I think you need to see this in its full. You go down the page, and look at this diagram here. Okay, the electrons will be passed down the chain. Yes. Electron is not equal to energy. Electrons can gain energy and it can be excited. Chlorophyll. Yeah. Correct. Yes.
Okay, then how is the glucose and the oxygen
Produced, right? Haven't come to that yet. So I just need you to appreciate what is happening at the photosystem.
So it goes down the chain,
Yes?
and it reaches this end point where there's too much energy.
What are you reading?
It's an arrangement of protein strings.
Or why are you asking that question?
It's like
much easier if you watch it in movement. Okay, so far I've been focusing you on these small little structures, right? And nothing beyond that. Okay? But if you want to, let's watch out where do the electrons go after it leaves here. Okay, so this is the chloroplast. And you can see light striking the chloroplast. Now let's go into the chloroplast. Here you will see thylakoids. Okay, the thylakoid membrane. If we were to zoom into the thylakoid membrane, that's where you see all the structures we'll be talking about. Outside the thylakoid, there's the stroma. So now let's go into one of these membranes. Have a look. On these membranes, you will find the photosystems. The photosystems are the one in green. You see? Yeah? In between the photosystems, there are just a series of proteins present. You see light striking the photosystems? Yeah? So the photosystems will collect energy. Now let's zoom into a photosystem. See all the circles over there? All those circles are chlorophyll pigments, absorbing the light energy. Do you see something being transferred from chlorophyll to chlorophyll? Okay, I go back a little bit. Looks like ping pong like that, right? You see, then it will start ping, right? Okay, that's energy being transferred. What happens as the energy gets transferred? This chlorophyll over there gets extremely excited. Okay, that chlorophyll over there is what I'm showing you at this point over here. It's getting very excited. When it gets excited enough, okay, it will lose electrons. Okay, once it gets excited enough. Simple, right? Okay. So then where do the electrons go after it gets excited? Okay, then you just pass it down the chain already. Okay, so now we zoom out. So just absorb a lot of energy. Okay, then electrons get transferred from protein to protein. Okay, so you get transferred from protein to protein. Let's zoom out a little bit more. Then you can see the entire thing in its full. Okay, and this is the entire process. See the electrons flowing from chain to chain. Yeah. But there's still a lot of blanks. I haven't reached all those points yet.
Wait, then what's the one with the special center?
Where is us?
The one with the reaction center complex.
Okay, the reaction center complex is within the photosystem.
So every single one is a reaction center.
Every green thing you see there has a reaction center. Okay, so if you want to look at your textbook, where's the reaction center? You look at your textbook, you see label over here, reaction center. That's what it's referring to. It's referring to this center part over here. Okay. Anyway, we are not complete. That's why it looks very incomplete. Okay? Okay, so can I get a volunteer to come out with me? Okay, Alpha, you help me with this now. Okay, Alpha, you will be this photosystem, I will be this photosystem. Just to let you know again, these represent electrons. Every time we jump out of the whiteboard means it's getting excited. Okay? Alright, so now I'm going to be this photosystem right now. Okay? So let's do it together. Ready? Okay, so Alpha, light has struck our photosystems. Let's get excited. Okay, excited? pass the energy on to the next electron, and the next electron, and the next electron, and finally we pass it to this center electron that gets so excited it jumps out and into the next protein. Following? Yeah, it occurs at every photosystem. Following up? Okay, but you see the problem now. Okay, alpha, right? Now the problem is this, Okay, you continue passing on the chain. Okay, you move, you pass on. Okay, where will I pass it to? Okay, sorry, you pass here. In cellular respiration, oxygen is the final electron acceptor, right? Plants don't use oxygen as the final electron acceptor. Plants use something called NAD+. When it receives electrons, it will become NADPH. You see what we're doing here? We're doing the reverse of cellular respiration. In cellular respiration, NADH gives up electrons. Here it receives it instead. Okay, we are just doing reverse cellular respiration. Okay. Now, notice that I'm missing an electron here now. Well, I can replace it with the electron from here. So alpha's electron will come here. Okay, now I have a problem. This photosystem is missing an electron too. Where can you get electrons from? Okay, then we can get it from water. Water will donate electrons into this space over here. So what we are doing here is reverse of cellular respiration. In cellular respiration, NADH gave its electrons, oxygen receive it, produce water. But in photosynthesis, water will give its electrons, and then NAD+, will receive it at the very end. It's reverse cellular respiration. Okay, thank you all. Okay, we'll take a pause here. Next week we'll continue and we'll look at this again. Is it recess now? Okay. Hey, thank you. You may go for a recess now.